All right. So thank you so much for joining us this morning. And what I want to do is introduce the team, first of all, who is responsible for operating and leading the biorepository. So I'm Norma Nowak. I'm the director. It's one of the hats I wear at UB. I'm also the senior advisor for life sciences at the Center of Excellence in Bioinformatics and Life Sciences and the Business and Entrepreneur Partnerships. Dr. John Tomasweski is the chair of pathology and anatomical sciences at the university, as well as the clinical chief of pathology at Kaleida Health in Great Lakes. Dr. Don Yerjo is the associate director of the biorepository. He also works with me as the associate director for the Genomics and Bioinformatics Core. Our newest member is Trevor Ralph. He is the biobank lab manager, and he is learning all things biobank these days. Most importantly, John holds the New York State tissue license, which enables us to collect tissues and store them in the biobank in the state of New York. All right. For some reason, John, that's not advancing. There we go.
There you go. A little slow.
So the biorepository has implemented a highly automated environment of technology platforms and workflow systems, ensuring all biobank services and facilities have minimal variation in quality and maintains the original integrity of the biosamples. This is a result of harmonized sample collection and processing and an increased accuracy of sample identity. It is an enterprise-wide resource serving the entire community here at the University of Buffalo, as well as our Kaleida Health and clinical partners, Great Lakes. We also serve our industry partners, so the young biotech companies that are present, and examples are Circuit Clinical and Neurovascular Diagnostics. It's located in the Clinical and Translational Research Center in room 6046. Don, I'm going to let you take this slide.
Sure. As with any essential biorepository, there are 5 main things that we can do to ensure that the samples that you store with us are essential for your future research program. So this includes a project consultation where we work on logistics and working with you from collection of the sample through storage and as well as distribution. We have a storage facility that has multiple deep freezers, -80 freezers, as well as a liquid nitrogen storage. And all of this is controlled by a monitoring system from ReScientific, which measures the output of the freezers, the temperatures every 5 seconds, and is stored and backed up within the biorepository. Another essential feature of the biorepository is sample processing, and this is in a highly automated, facility, which includes multiple robots from Hamilton, which can do both blood fractionation as well as biosample aliquoting. We have the Revity chemogen system, which can isolate nucleic acids from a variety of tissues and samples, from saliva all the way through whole blood. And this is from as little as 200 microliters up to 10 mLs in total. The most essential part of the biorepository is our quality control and analysis. This includes nucleic acid quantification through the Agilent tape station, where we can determine the qualification of each of the samples visually, and through the Lunatic, which can determine both biosample concentration and quality, not only for nucleic acids, but also for proteins such as serum that are located within serum and plasma throughout the process. We also have a molecular barcoding instrument from Standard BioTools, which can determine SNPs, indels, and also from cancer panels, whatever mutations are present in the samples that you are storing with us. And all of this is controlled under our IT infrastructure through our SLIM system, which is currently being implemented, as well as a new quality document system named Qualitize, which is going to be implemented university-wide. Next slide, please.
So as many of you know, one of the reasons we actually established the biobank was to try and pull all of the biosamples together in a centralized resource. As scientists, we like to keep our samples close to our vest and in our labs and in our freezers because it's convenient. But one of the issues with doing that is when every single lab actually takes a biosample, stores it, prepares DNA, RNA from it, you're losing that ability to standardize those processes. So what we're able to do is to really make sure that the biosamples are processed in the same manner, eliminating that variation, and in an automated way, eliminating mistakes such as mixing up samples. We really want to shift the culture that we have here at UB from this many silos of samples in all of these different labs to centralizing them in the biorepository. We're happy to make working copies so you can still keep a copy in your lab, but keeping the human biosamples in the biorepository, I think, is really central to being able to use them for many years to come, as well as to collaborate with those samples and create consortia, as well as work with our industry partners. And remember that samples really aren't meant to spend their entire life in a freezer. Their purpose is to produce data, and think of each sample as a biolibrary, which really allows us to make new discoveries and ultimately improve the health and well-being of our population. So this is just an example of the types of processes that can be performed on the different biosamples. And we have different technologies in the genomics and bioinformatics core, as well as in the biorepository itself. But what you're looking at is a network with the four corners of omics technology, of omics: the genome, the transcriptome, the proteome, and the metabolome. And the processes that you see in the center, many of those are just technologies that we have across the university, from mass spec to next-gen sequencing. And in John's pathology labs, they use image as a very powerful technology. So what we're hoping to do is be able to interrogate each level of the genome and have an understanding of the power also of evolving in new technologies and making sure that those samples, whether we use them today, a year from now, or 10 years from now, are available for those new technologies to interrogate them. And one of the technologies that we're currently building here at UB, and it crosses both John's cores in the Department of Pathology, the Biobank, and the Genomics and Bioinformatics core, are spatial omics. These enable the localization of transcripts and proteins at cellular and subcellular levels. And I'm going to let John expand on that.
Thank you, Norma. So when you look at this diagram, there is a wide variety of omics, multi-omics, to interrogate what in essence is a disaggregated cell and tissue mush. So the problem for investigators is to translate those distributions of molecules to the actual functional part of an organ, which is the cell and tissue network. And this concept of spatial omics basically adds location to where the molecules actually are doing their business. And the fundamental posit being that you can't understand a systems in a system biology way unless you know where those molecules are interacting. So it's about cells talking to cells. Next slide. Yeah. So we in the collaborative cores in the School of Medicine and actually in health sciences have been working hard to create deep annotation systems using cellular and tissue imaging tools to understand where those molecules actually reside. We have capacity to do standard histology in a computational and digital way. We run an image server for all those optical data that come out of a variety of machines. We are pretty well equipped both in the School of Medicine and across the health system with multiple both wide field and super resolution technologies. We have STED, we have fluorescence lifetime imaging. We have a variety of high advanced technologies. In addition, in the most recent past, we are starting to adopt technologies that allow us to put highly deterministic labels onto these images. So 3 weeks ago, through a lot of support from Cummings Foundation and a variety of other sources, we've taken on the Standard BioTools Hyperion, which is an imaging mass cytometer, which allows us to do very, very deterministic 40 channel multiplex labels on images and do validations of these data that you get from disaggregated investigations. All of this is to say that the real value in the biosamples isn't having something in a freezer. It's having the annotations, the data attached to that something in the freezer that really matters. So it's well annotated, high quality, reproducibly processed materials that really drives innovation. So all targeted gene therapies, personalized medicine, really require well annotated biological samples. If you don't have the consistency of those annotations, you don't have the consistency of the pre-analytics, you're not going to be able to do coherent science. So this growth in omics technology, diversity of sample types, really fits into the biobank structure, where the biobank can actually provide the visibility of those annotations to support whatever you want to do, sample selection for your research project, improve your utilization, as Norma mentioned, prevent duplication of disparate collection techniques, and then and always have that available and exposed. And so our SLIMS, our laboratory information management system, that is exposed through through the university, allows investigators to see that. So understanding the quality of the specimens, understanding the tags or annotations of the specimens, and being able to see them is sort of critical to advancing science on our campus. Don, do you want to talk a little bit about sort of the access and logistics of all this? Oh, one more slide. Sorry. So the ecosystem that we're talking about here is sample collections, patient sample collections, experimental design sample collections, and the biobank doing something with that, not only taking it in, but actually extracting materials, understanding the quality of those, documenting their consistency, and then utilizing core laboratories in UB to collect additional data that is needed, but doing it with a consistent pre-analytic sample that allows minimization of the variance on those materials. Now, Don. Thank you.
So why, just to kind of summarize, why would you want to use the biorepository? Well, obviously a number of factors play into the role of the biorepository in your research studies. The main one is that research dollars at this time are getting more scarce, and in order to remain competitive, as well as to make those research dollars stretch a little bit further, we have the infrastructure already in place currently at UB to allow you to access both samples that are archived, as well as the automation and the infrastructure to support research, where you don't have to purchase freezers, for instance. We can store them here in the biorepository for future studies as well. We have a GLP facility, which allows us, or a GLP compliant facility, excuse me, that allows the samples to be utilized for downstream studies and distribution to other facilities, collaborators, internal researchers, etc. This allows our automation platform to process the samples in a compliant manner, as well as to ensure that the data that is generated from those samples is of high quality and for use as for both research and clinical applications downstream. And all of this can be distributed to collaborators as well as additional academic researchers throughout the UB campus as well. And I believe it is essential to have these facilities to ensure that we maintain our research status here at UB. So how can you use this services? Well, you can contact us. Reach out to Norma, reach out to us. You can look at our website, as well as send us an email that we have on the first page as well. Next slide, please.
Okay, there we go. All right, so this is our faculty panel, and they're going to speak to their experience with the biorepository and the value it brings to their research programs. Our first panelist is Dr. Adnan Siddiqui. He's the vice chairman and professor of neurosurgery. He is also the CEO of the Jacobs Vascular Institute. Dr. Siddiqui, are you here?
Yes, I am, Norma.
You're wonderful.
Yes. Well, thank you very much for this opportunity. And it's great to be in partnership with John and Norma and many others. I see some familiar names on the list who are working with us. And I'll briefly tell you what we were interested in and where Biorepository has been absolutely critical and pivotal in our being able to achieve success, which is manifested both in terms of a large number of publications, but also actual funded grants, which continue to accumulate based on this incredible resource that we have actually used quite effectively, including recruiting faculty together with John to really sort of harness the power of this endeavor. So I'll give you a brief update as to what we are looking at and what we're doing. Basically, we are trying to collect biological specimens in conventional and innovative ways. So we are doing both. So for example, we remove clots when somebody's having a large stroke from an embolic source or atherosclerotic source, and we get those samples. And previously, they would look great on the table and say, wow, I got a clot out, and then it would go into the trash. And if we just send them to John's clinical cohort, we would get the answer back saying it's a clot. But now, based on the incredible work that Vince Tutino is doing, we are employing AI to characterize the overall shape. We are doing histochemical analyses. We're doing proteomics, genomics. We're doing all sorts of stuff to understand where these clots are coming from, possibly to accelerate secondary prevention of a second episode by telling beforehand, this came from your left atrial appendage, so almost certainly you have paroxysmal atrial fibrillation, et cetera. So, or it came from your leg vein, which means there is a PFO. We are also collecting specimens for GBMs, and that's another exciting part of the work we're doing because, well, there's a lot of brain tumor banks out there that collect GBMs, but what we are doing is inoculating rats with nude rats with these tumors to assess the value of intra-arterial chemotherapy for these incurable cancers. So I think that's really exciting to be able to do it. And again, incredible partnership with members of John's team, where we are creating now organoids based on human GBMs that we have harvested during surgery. And that's fresh off the press. We are actually working on our first sets of functional organoids to create implants. We are also, this is longstanding work that Vince Tutino started, collecting blood specimens from people who come in for diagnostic angiography, simply arterial blood from their peripheral vessels to create a blood test. 5% of the US population has brain aneurysms. So it would be wonderful to know which ones we need to follow if we had a screening test that could be performed. So that's a big part. And then every now and then when I'm actually going to the operating room to actually clip the aneurysm, we're harvesting tissue directly from the aneurysm, an inachievable sort of sample by harvesting it. And again, doing all the proteomic genomic work on that. Again, Vince is a key part of that. And finally, we came up with a really cool way of trying to get specimens, at least endothelial specimens, where when we are treating these diseases endovascularly, we can advance coils and deploy the coils in the aneurysm sac or whatever structure we want, and then recapture the coils and have enough cells stuck to the coils that we can then harvest and perform single cell type functionalities or assessments. And we're doing that for aneurysms, we're doing that for AVMs. We're really excited to maybe start doing stuff with intracerebral hematomas and subdural hematomas, and maybe expanding the brain tumor program to other tumor types, meningiomas, et cetera. So there's a lot that we are doing in every domain, and it's impacting our clinical work and our translational work, and even our very much basic investigational science work. So we're delighted that we have done, and continue to look forward to doing more. And I'm happy to take any questions if anybody has any based on my experience thus far. Thanks, Norma.
Thank you for joining us, Dr. Siddiqui.
My pleasure.
Dr. Pili. So Dr. Roberto Pili is the Associate Dean for Cancer Research and Integrative Technology at the Jacobs School of Medicine and Biomedical Sciences. Roberto and I go way back to our Roswell Park days when he came to you, left Buffalo, went to Purdue, and I told him I would never forgive him. And then he fortunately came back. So I have forgiven you, Roberto. He's going to speak to us today on building a oncology-focused biobank, and really right now he's focused on genitourinary tumors as a pilot.
Good morning. Thank you, Norma. And thank you for the invitation. Thank you, Jill. It's a pleasure really to be part of this team. And again, as Dr. Siddiqui was mentioned, that we could not do this really big if it was not for you, Norma, and John, and Don. So that has really been a tremendous effort. Is really your effort is, your bank is really a jewel at the UB, and I'm glad to see that this is come to fruition, and more people actually take advantage of this platform. As Norma said, that actually, Norma, Jill, you have also the next slides. There is a yellow box, but this has been an effort that's been going on for quite some time. So thank you, Jill. And so we put actually our first patient enrolled in this protocol 1.5 years ago. And to date, we've enrolled actually probably more than 190 patients. And we collected probably more than 1,200 samples. It started, as Norma says, with GU cancer, so prostate, kidney, bladder cancer. This is our research interest, but now is open to any solid tumor patients. So any patient that walks in through Great Lakes Cancer Care can be enrolled in this biorepository study. So we have access to archive tissue, and of course we try to get a fresh biopsy or a fresh sample from primary surgery. And I think the neat part that, for example, differentiate this protocol from Roswell Park Biorepository, at least for the general repository, is really this prospective blood collection. So every 3 months or 6 months, when the patient come to our clinic, we collect blood. And so we collect the serum, plasma, and PBMC. So this is a tremendous platform for, you can imagine, for drug discovery and drug development. We're particularly interested, of course, for GU malignancy, as Dr. Siddiqui was mentioned. We also established this 3D culture for drug screening, and we try to establish actually patients that are xenographed. This is just a short list of the GU PDX that we established, more to come. And again, this is a tremendous platform that, it's still small, it's about 190 patients, but it's meant to grow with additional resources. And this is really meant to be open to all the UB investigators. We have established the UB Cancer Research Consortium is across 20 different departments, across 8 different schools here at UB. Again, this is really open to any investigator at UB. I'm planning actually to have, eventually my dream is to have a SUNY SUNY Cancer Research Consortium. So to also have our partners institution to have access to this biobank and to see whether we can join the biobank for cancer across the SUNY system. Again, I want to thank again Norma and John and Don for this effort. Again, we're very excited about this availability. We're growing, and we want to make this really available to anybody interested in cancer research. And I saw here Wilma Hoffman. Wilma, we have a small project together. We're trying not to identify now the patient. She has a cool biomarker for potential for prostate cancer. And again, this is a really a biobank meant for research. And as Norma, you mentioned, there is no reason to collect samples if nobody's using it, right? We're here to collect and use these samples to advance science and to advance research here at UB. So thank you for your attention.
Thank you, Roberto. The other thing I should mention, both Dr. Siddiqui and Dr. Pili are also expanding through organoids and spheroids to make sure that these samples aren't exhausted. So they, again, renewable resources for the samples so that we never run out of them. Okay, our next speaker is Dr. Matt Bonner. And Matt really was another driving force for establishing the biorepository several years ago. Matt was awarded a large grant to look at environmental pollutants in the area around the Tonawanda Coke factory. So I'm going to let Matt pick it up from here. He's an associate professor in the Department of Epidemiology and Environmental Health in the School of Public Health and Health Professions. And his story is really a tremendous one. And without him, I think the biorepository, he was really our first big project. So he can speak to how we've enabled him and the things we've learned from him.
Thanks, Norma. Excuse me. Thanks, Norma. Good morning, everybody. Yeah, so I've been involved with the biorepository for a number of years now. And as part of the environmental health study for West New York, excuse me, as Norma said, which is a large prospective cohort, we have over 13,000 adults from Grand Island, the city of Tonawanda, and the town of Tonawanda. But as part of that, we've got a sub-cohort of 2,000 adults that we've collected biologic specimens from. And then 1,000 of those individuals have also donated a second set of specimens. So we've collected urine, we collected 2 trace element tubes, 2 EDTA tubes. And then with the pandemic, we switched to mailed collection and started collecting saliva. So from the urine, when we worked with Don and the biorepository crew to put together our protocols so that when we ship it to them, they go through their protocols to handle it, process it, and store it. So for each participant, we got 24 aliquots of urine for about 69,000 1 ml aliquots of urine. We get 6 serum aliquots. We've got about 8,000 of those, about 15,000 plasma and buffy coats, and then 839 saliva samples. So we've got about 94,000 aliquots that are being stored at the biorepository. And it's really been very smooth processing for us on our side. It's been a relatively headache-free process overall. A couple of years ago, we did an audit of our samples to make sure that what we thought we had is what was in the biorepository, and that worked out really pretty well. And so then from there, what we do is we contact the biorepository and I'll do this every couple of months, and I'll ask them for a set of urine samples, which is what we're analyzing right now. And they'll put together the batch. We've got some QC aspects that we want particularly done for our study, which includes blind duplicates. About 10% of all the samples will have blind duplicates. And so I've worked with Don and the team there to make sure that that's done smoothly, so that the laboratory that receives our samples can't tell the difference between the blind duplicates and the actual participant samples. And that's worked out really well. The research team receives their invoice, and they link them with aliquot IDs, and it's worked really pretty smoothly overall. I think the big advantage for me as an investigator has been that it's relatively headache-free. It's been a good experience overall. I don't worry too much about these samples once they leave us. And so it's been a very good experience overall. Thanks, Norma.
Thank you, Matt. And one of the things we wanted to mention too is it doesn't matter what campus you're on. We have couriers which will come and pick up samples. The biorepository team will also, we don't just sit in the biorepository downtown. So we will do everything we can to facilitate the transfer of your samples to the biobank. And our last speaker is Dr. Vincent Tutino, who's an assistant professor in the Department of Pathology and Anatomical Sciences. Vince also is, I believe, a co-founder of Neurovascular Diagnostics, which is a young biotech company here in Buffalo. And he collaborates extensively with Dr. Siddiqui and the Cannon Stroke and Vascular Center. So Vince, I'm going to get you all going here.
Yeah, you can probably just go to the middle slide, the next one. But yeah, thank you for having me. I'm very glad that Dr. Siddiqui spoke first, because he pretty much covered everything that I do as well. But I guess the things that I want to point out about the biobank, and this is kind of like our whole pipeline here, at least for some of the liquid biopsy work. The thing that I would like to point out is kind of in between the sample collection and the extraction, and in between the extraction and the sequencing, there are a lot of QC steps that go into all of this. And I think having all of our samples at kind of one central location that all go through the same process that is continuously monitored really helps with that. You can see here at the bottom in the studies that we have in developing blood-based RNA biomarkers for aneurysm, we have 500 patients samples, and it keeps growing, especially with a new grant that we just got that's going to extend this work by another 3 years. And so when you're dealing with that many patients samples, it's really important that we have a baseline level of quality control and a way to analyze it. And I think that's one really important thing that the biobank offers. Other couple of things is, going between the sample collection to RNA extraction to sequencing and analysis, the biobank and the sequencing core kind of offer this centralized hub to do this almost all under one roof, right? So you're not worried about independently checking out samples and you yourself passing them from one entity to the other entity. There's kind of this seamless transition where there's oversight across the whole process. And so that has been very beneficial for us as well. And the last thing that I want to mention, at least for my lab, has been a sea change in kind of how the research is done in general, almost, facilitated single-handedly by the biobank. When I was doing my PhD and when I first started mentoring students to assist in all of these studies, we were doing a lot of the processing by hand, right? And so it took up a lot of time and a lot of manpower to do this. And now with some of the capabilities that Dr. Tomasweski and Dr. Ujo were talking about, especially with the Key Magic 360s, a lot of that is kind of taken out of our hands, which, you know, for the students, they don't get to play with the samples on the bench, but for us, in charge of the studies, it is really, really beneficial to see kind of standardization at many of these junctions, right? And that actually enables us to hone in and focus in on other areas of science. So, you know, a lot of my students now, rather than spending lots of time learning some of these benchtop techniques, they're focused now on learning computation, right? Because this expression analysis, bioinformatics, and biomarker development, you know, portion of the study, they can now focus on doing that. And so we're building, you know, people who have a biological background, but also are computationally focused, which is, you know, one of the main goals of the pathology department's CCBAP program. And so this is kind of facilitated by John's vision with the biobank and everything. So yeah, if there's any questions, I can, for me, I'm happy to take them, but those are just, you know, three kind of important points that I'd like to highlight.
So Vince, the other thing that I was very impressed with is your acknowledgements and how the biorepository has really served as a hub for collaboration and the groups that you've brought together for these projects. Yes. So again, think of it as the place where people come together, build consortia, you know, build teams across UB and outside of UB, and especially for this new initiative that's going on called SUNY Strive. Teams of consortia and cross-university sharing of resources is going to be so important. And this is just a great example of that happening within, and now we'll be going outside of just UB. So thank you, Vince. That was a great few minutes of your time, as well as, again, you've done so much with the samples, and I think you and Dr. Siddiqui have built a wonderful program.
Thank you.
So does anybody have any questions that you'd like to ask the panelists? Wilma.
Yeah, hi. So this was great. I learned a lot about you know, the different parts. I think my question is, are there any, so right now it seems if you're a part of a group that collects the samples, you have easy access. Is there anything are there any plans to make it accessible for like all of the UB community? Are there any plans to standardize access to make available all the samples you have? So if I want to go now and work on a blood clot or something like that, is there anything in plan that I can look at the list and say, oh, I want to have those patient samples and work on those?
Don, you want to talk to the SLIMS access?
Yeah, so one of the things that we're implementing here is a laboratory management system, so that once we input the samples and the aliquots into the biorepository, it is then becomes part of our database management system, so that then once those samples are stored, we'll have a forward-facing access point for PIs to look at what samples are present in the biorepository. So then you can search under, let's take an example for if you wanted to look at clot samples from patients who are between the ages of 50 and 75, for instance. So you'll be able to access that kind of information once it's integrated with either REDCap or within our own systems. And this will include RNA sequencing data. It potentially could show images of the clots from H&E stain from pathology services, as well as additional information, molecular barcoding, SNPs, mutations, etc., that we can implement there. And that would be able to be downloaded by you and then used for whatever purposes you can. We can also set it up as well with integration with iLabs in order to set up an invoice where we can then retrieve those samples and then deliver them to you through logistics, as well as just courier services or some other type of delivery system.
Wilma, the iLabs project is just being sort of fiscally and administratively put in place. The pilots will occur in Q1, Q2 of the next year. iLabs is a suite of core facility management tools. The Biorepository actually was the first on campus to put in one of those iLabs modules, which is called SLIMS, which is the Laboratory Information Management System that Don was just talking about. But there's a whole bunch of other fiscal, inventory management, asset management, quality management tools in there. It is clearly the best of class for all high-performing universities, and we're very excited to be, piloting this in the next calendar year. So more to come, but I think the answer to your question is, yeah, that development is underway. You should be able to actually look at this stuff sometime in 24.
Thank you.
Wilma, if I may, and I apologize, I need to jump off, but I think you were alluding also at probably a tissue utilization committee. We're talking with Norma. Of course, right now we have plenty of samples and we don't have many investigators using it. Once we're going to have more investigators using it, of course, we're going to be careful how we utilize these samples. Right now, again, as far as the cancer tissue and cancer-related samples, I mean, these are UB and belong to the biorepository. And of course, if people wants to get access to this, they go through Norma and John and Don, and with my assistance, we decided what type of project we do. The same things I assume with Dr. Siddiqui for the neurobank. As we are growing, we're going to establish this tissue utilization committee like Roswell Park has. But the challenge, this is a challenge, but also opportunity. Again, those committees are going to be not there as a gatekeeper just to preserve the sample, as to really to allow anybody, like you said, Wilma, to utilize this sample. And my goal is eventually to expand the access to this biobank, not just to UB, but throughout the SUNY system. So we're going to have more and more investigators ask for these samples, and how we can regulate the access to this sample. I think that's something that, of course, we need to still look into it details. But the goal here is to collect sample and to use it for research.
So I think both John and Roberto too, I think a lot of our investigators are going to be looking for samples which have certain biomarkers in them. So if something, John, has been tested for, like, let's think about lung cancer in the ALK gene. Mm-hmm. That would be information that potentially it's not PHI, but we could store that in.
Right. So these are the annotations that I was speaking of. They could either be single annotations, as you just mentioned, or they could be multiplex annotations, which actually allows you to start to do all that machine learning and AI that everybody has a buzz about, discover patterns, connections that maybe are not apparent to you. That's the real power of this. So when a biosample is not a piece of tissue in a freezer at all. What it is a dense set of data attached to that, and attached to maybe the clinical record if you have IRB permission for doing that, that allows you to actually do something. So the biobank is not a freezer farm.
One additional comment, if I may, as this biobank is flourishing, I think it would be great to have if you think about also as a service, you can imagine how to regulate the service, but in terms of also of sequencing, and more importantly, bioinformatic support and analysis. That, as you know, this is a major challenge that we have at UB, the bioinformatics. But it would be great as you are raising funding for this project to secure funding also not only to store these samples, to collect and dispense, but also to analyze this sample and to provide the full support of investigators that are utilizing this. So when I'm trying to say that, investigators that utilize a biobank sample, it would be great if they can have a support also to sequencing those samples and to analyze those samples through your core.
Dr. Tutino's SBIR phase 2 that was just funded has in it a big piece of utilization of the sequencing core, utilization of the biobank, and Vince, I think there's a couple other cores that are built into that that grant. So that's exactly, Roberto, where we're going
Perfect.
with all this.
Great, thank you.
So if I could ask a question, and first of all, before I ask, I think I want to just mention on behalf of the university a sincere thanks for Dr. Tomasweski and Dr. Nowak for bringing this resource to fruition. If you look across many very competitive research institutions, this is an essential element to supporting research programs and core investigators. So they did a remarkable thing in bringing this to Buffalo, and so thanks to them. I just also wanted to ask either of you, what are your thoughts for growth, or what are your thoughts for additional elements, and what support do you need in order to make that happen?
Well, if I could begin to answer that by answering Dr. Pletnikov's question in the chat. He asks, are there immediate plans on expanding collection to include postmortem brain samples for molecular neuropathological studies? Yes, that's under discussion. Those kind of retrievals are complicated. There's a whole intersect with the anatomic gifts program that has to occur, and all the logistics of actually doing sort of warm harvests, if you will, that allow you to get decent quantities and qualities of RNA and do all that spatial omics stuff that we just talked about. So it definitely is high on our list. We are going to need, though, to be creating collaborative groups. These are complicated systems that, with funding, that allow us to do that. But we do have the infrastructure in place. We have a robust anatomic gifts program. We certainly have all these technologies that we just mentioned. We have this biorepository. We're set to do this kind of stuff. And I know Dr. Pletnikova was instrumental in doing this at Hopkins. And I think, kind of taking a page out of that playbook would be really advantageous for us to execute on. We're also very interested in not only doing the postmortem collections, but actually crafting interstitial pluripotential, induced pluripotential stem cells from skin fibroblasts, for example, and developing organoids from those donors that could be immortalized and then related to the molecules that we collect from the brain harvests. So a lot of exciting stuff going on.
If you have any other questions.
Does Wilma have her hand up again?
Yes, I have another question.
Yeah.
And that is, so we're currently trying to work, it's not technically a biobank issue, but we are working on a project where we would need a live tumor tissue to basically treat the cells live. Right. Is there anything, and I've talked to several people, it seems one of the big issues is to find somebody to actually collect the samples.
Well, we've started to do that. We've started to do that with Dr. Perry in lung cancer. In the last 2 months, I've done, I don't know, probably about 10 of these collections. It is a lift. I've been sort of pushing this myself, and extensibility is and resource is always an issue. I know you're interested in prostate. We're trying to gear up to do our first prostate collection under Dr. Pili's IRB. Prostate in particular is a difficult collection because of the surgical pathology necessity to maintain capsule. So capsular incision is a big deal. If we can't stage that tumor, it's a big deal. Yeah, we're kind of gearing up to do our first one of those. So more to come, but we actually have started on this, Wilma, this in the past quarter, basically. So it's been a long time coming, but we got started.
So Vilma, are you looking to make organoids, or what would be the purpose for the live cells?
So currently we're working on a lung cancer project where it seems as we would need to treat the cells to certain metabolic conditions to see how they react. And it seems they react differently. So I'm not trying to make organoids. The samples probably won't be usable later.
Okay.
We would just need piece
So
of
you need them
tumor.
Dissociated?
Pardon me?
You're going to dissociate the cells, or you just want a piece of tumor that you're going to put in culture?
Yeah, we have protocols where you can isolate the cells and they'll grow, whatnot. But it won't be usable. Pardon me? In culture,
primary cultures, yeah. We've been doing this at BGH. So within the hospital facilities, we actually don't have the biohazard hoods to actually do a sterile collection off of a specimen. So right now we're bridging it by, me personally taking it to the biorepository and personally taking it back. But I think there's a discussion in Kaleida of trying to put those facilities in place. So as we get a little bit more, endowed with equipment and facilities in the hospitals on the clinical side, we can do that translation. That certainly is where my background is. So I know where we need to go. It's just been slower than I had hoped.
Okay, well, if there aren't any other questions, you have information on how to reach each of us. You can also go to the Biorepository website, which is going to be updated. There's information there currently, but please feel free to reach out to myself or Don Urjo. And even if it's just to have an exploratory conversation, we're here to help you and support your research project. And I think if you really look at the bigger picture, the Biorepository is like really the beginning of the path down translational research and commercialization. So that sample, again, contains tissues and cells and structures that can be used to, again, develop tests, diagnostics, and potentially new therapeutics. So without it, none of this stuff would really happen. Tissues are the heart of translational research. So please work with us, and we'll do everything we can to let you focus on the research in your lab, but provide you with high quality materials with which you do that research on. So thank you again for attending. Jill, thank you so much for joining us. And for my wonderful panelists, thank you so much for the work you're doing and working with us in a collaborative manner.